Okay, thank you all for having me talk today and having the CASS crew put up with my procrastination. So today I'm going to talk about mainly AUC and viral vectors from R&D to GMP. So some of the things I want to touch upon is before we go into the in-depth AUC analysis and how this can be applied to in R&D characterization and also into the GMP environment, I want to talk a little bit about the role of biophysics in gene therapy. Now, please remember, this is going to be a very broad topic, very general, 10,000 feet type of seminar. So if you have very detailed questions, just send me a link or ask questions later. So what do we have usually in traditional biophysical approaches? So we usually have DLS and AUC for to looking at higher order capsids. By the way, higher order capsids, higher order structures, and aggregates are going to be used interchangeably in this talk. DLS non-quantitative but very qualitative. AUC quantitative. Usually you have your size distribution for your low order capsids, empty, partial, full, and HOC. So again, AUC is being used at this point in time to do a lot of these type of experiments. SecMols, on the other hand, can and has a role in some of the characterization, but you have to be very careful with some of the data that is being produced from SecMols. For example, it doesn't capture your partial capsids. If you have overfilled capsids, it will not pick it up. So there are very some limitations that you have to take into consideration. Now, other techniques such as CIEF, cryo-EM, and TEM, and then most recently we have the mass photometry. So these techniques also provide some really good initial estimations of your MTs and fulls, but again, suffer from the resolution that you will get with AUC. Easy to use, they're very easy to use. However, well, probably not cryo-EM that much, but it is very good for the initial pass to get your size distribution. It does miss some information, but you can always back it up with AUC. And lastly, I want to talk about some of the other approaches, such as AEX, this wonderful technique as charge distribution mass spectrometry, which I kind of like also, but does give you some information on your partials and empty and fulls. And then you have specific chromographic techniques that you can use. But this is a disclaimer. Most of these techniques and these characterizations are all confirmed by AUC. So at this point in time, we know that AUC is the gold standard. So let's move on into that. So this is a pretty common CQA of the AAV system, very general overview. And you can look at many of the characterization methods you can use, including for purity and so forth. And AUC has a very pivotal role in not only characterization, but in the cGXP environment. And we'll talk about how that transition can be done and why it should be done. Because if you're presented data that is incomplete, then we're going to have a little bit of a problem. The regulatory bodies are going to have a little bit of a problem. So I want to stress that AUC gives you the most complete picture. And another disclaimer is that the only disadvantage of AUC, I see from my standpoint, and what I've been hearing, is the complexity of the analysis. Well, the complexity of analysis can be easily overcome by education and training. And that for me is not a limitation. You get trained, you can do this. So moving on. So usually empty partials, fulls, and AUCs can be done very easy with AUC. You have no dilutions, no labeling, no nothing. You can figure it out as it goes in, i.e. it's a first principle based technique. Now, some of these LODs you can get, in my hands, I can get LODs at 0.5 to 5%. And then you just multiply that by 3.3 to get what you you do for your LOQ. There's no modifications or buffers. You are seeing your sample at in its purest form at a higher volume than some of these other techniques that you have. So you can also figure it out from just the partial. Now, where does AUC come in? Do you need a purified sample? No, you actually do not need a purified sample. We at Bioanalysis have developed a method that you can use from your cell harvest. Yes, your cell harvest, and all the way down to your final DP. And you can track that without any special labeling, without any special buffering systems, just a routine AUC. It's just you have to do some extra little things, but it's literally an hour of work before you can do it. So AUC, again, can not only span from your cell harvest, yes, your cell harvest, all the way down through your process development purification, all the way to your final drug product. So what are some of the other few things you can do with AUC? AUC can look at your DNA capsid ratio, the extinction coefficient, and most importantly, your higher order characterization. I want to talk a little bit about the DNA capsid ratio and extinction coefficients, because this is the standard of what we do in AUC when we're looking at, for example, a stoichiometric interaction between A plus B. We have to figure out the extinction coefficients, and that's very easy to do with AUC, doing by the multi-signal approaches. Another thing you can do is the DNA capsid ratio, because the signal that you're getting at 260 or your interference up to 280 and 230, you can use that discriminatory power to figure out your DNA capsid ratio. Now, you can fine tune it by having some degeneracy in your wavelengths, but again, just to have an approximate estimate of your capsid ratios is pretty powerful. And I put up some equations here because as biophysicists, we kind of love equations. It's basically solving the Beer's law equation simultaneously. In this case here, there's a wonderful app note by Tom Lowy that talks about the power of interferometry, interference optics basically, that you can exploit to figure out your DNA to capsid ratios. You can help with the characterization of the partially packaged genomic capsids. And as we know, as the field is evolving, we're starting to get more into what is your partials? What are these extra things that are coming along with our final full capsid? So we have to have a good handle on that so we can really move forward. In addition to that, I've been talking very rapidly in this case because I only have 20 minutes. What can you get from AUC? Well, you can get capsid quantitation, AUC characterization. Do you have multiple species that are close to your full capsid that are not partial? So this is called an intracapsid heterogeneity analysis. Yes, it is stability indicator. And I'm not only talking about just looking at your HOS or HOCs. I'm talking about what is happening at every capsid. What happens to your partial? What happens to your full? What happens to your empty? What happens to the VP aggregates? You can measure that, and quantitate each individual species in the AUC as a function of temperature and time, and you can get very, very good data. In terms of, I just talked a lot about DNA capsid loading. There's a wonderful paper by Brenda Barnum that stated that that figured out that the length of DNA that you insert, your packaged genomic DNA, is linearly correlated with your S value. So you can tell overpacking, partials, you're full, just by having a good grasp on what you have in ITR, from ITR to ITR. You do not need extinction coefficients. You do not need any information a posteriori to figure this out. You can do it by and from data. Contaminants. We see a lot of contaminants in our species, whether it be a very large contaminant in terms of its size of the empty capsid, or in certain cases, we can see proteins, like small proteins in there, 26 kilodalton, that's floating along with that came along with your purified product. We can figure that out just by a single experiment, and then followed by another experiment in which you just characterize that. So just how you do in that cases. And this again pushes the graphical representation of what I've seen. I've modeled this data to show that you have a lot of HOCs in there. One of the things we have to get away from is that these are non-antibodies. So the HOCs are not going to be dimers. It's not going to be obligate dimers or anything like that. They're going to be mixed particles that you're seeing, and they can lead to polymers. It can lead to more larger quaternary structures that we don't know. So you have to have very careful consideration of that. A couple of things we have to think about in terms of experimental design in this case. So I modeled some data. This is what we usually think about in terms of empty capsid and full capsid, but in reality, it looks like that, a smear. So what new things you think really consider, especially when looking at higher order capsids or HOSs, you have to think about the road to speed. What are you doing with the road to speed? How is that affecting your data analysis? Spinning at a higher speed can produce non-ideality. You can have boundary collapsing. You can have non-laminar flow, okay, and have turbulence. Now, the Lamb equation does not help does not work well with turbulent flow. It works well with laminar flow. You can have non-ideality. In terms of your assay concentrations, what are you really seeing? Now, remember that the goal, the monetary unit, the prize position that you get from an AUC experiment, so an orientation velocity experiment, is your S values. Now, in the XLA and XLIs, these are really good, robust values. There is a little bit of, problems sometimes with the optimum AUC, which can be fixed. And there's a great paper by Lowy and Berkowitz that talked about this. But usually your S values tell you a lot of information. So you have to be very consistent in that type of approach to your AUC experiments. Now, some things you talk about when you spin fast, you want to improve the resolution, but sometimes improving the resolution doesn't always improve the fit. And this is when you have low species in there. So very careful attention has to be done. Now, given all of this and this complete package, once you have figured out what is the best fitting parameters, the AUC experiment is so robust that those samples and that fitting parameters will always give you the same result. It is. And we have gotten percent CVs on quantitation less than 5%. We have gotten percent CVs on S values less than 0.3%. That is precision there. I mean, very precise numbers. And with this precision, knowing what we have, it's just easy to put it into the cGMP environment once you have the correct things. And some of those is, how do you do this first? Before we get into that, let's talk about that analytical landscape. So I use SetFit a lot, and there's multiple programs you can use out there, Ultrascan, SetAnal, DCDT, and so forth. But what we try to do is maximize the best and easiest visualization of the data. And this is basically, what are you doing in the method development? You're looking at orthogonal modeling, signal to noise evaluation, and experimental design. That should give you your first pass at your method. And that is what we're trying to do. Now, be very careful. I'm going to give you an example right now that will kind of shock you a little bit. This is a data in which it looks like random noise, okay? We can fit that data. You see boundaries that the eyes can't see, but it will give you some information. Now, is that information correct? Probably not. So, you got to be very careful on how you fit the data and what is your concentrations that you're using. What is your regularization? What is the best one? Max entropy, know the limitations of it versus second derivative. Again, know the limitation. The identification of artificial peaks. Are we in a local minima? How does this stack up to different types of modeling? And once you have that locked, it really doesn't change unless the sample is changing. And that's what we are trying to push here. Now, the biggest complaint is usually 21 CFR, and I'll talk about that in a second. Here's a good example. Know the limitations of maximum entropy. Here is a good data. I model this data, so I know exactly how many species are in there. Okay, but you look in there, it looks like a smear. What does the alternative model look like? It looks like a smear, but does the concentrations add up to the same thing? Does it give you the same values? Well, if you do a little bit of optimization, you can get that blue line there to give you nice discrete species. You can further optimize that by knowing what the programs are doing and really implementing that into your analytical modeling. So I'm going to try to talk a little bit more about AUC functions. So basically, going from that method, and you know what you have, you can, again, you emphasize that method development. There are multiple analytical responses. So you're not only looking at your empty capsid, you can look at your full, given if it's in a certain ratio. You can design cGMP methods that are not only looking at your percent empty, but also your percent partials or even your percent HOCs. You have to be very careful. So emphasis on method development. Then you go into the qualification. And when you're in the qualification, it should just be a checkbox. One of the biggest problem is the 21 CFR compliance. There's going to be some things that are going to come out in the future that will change this. Currently, Bioanalysis has a method for that. What we do is that we have our custom capsid integrator. You're not using a program that was built for something else to do the integration and give your averaging and so forth. We have built an Excel program that's 21 CFR compliant that you can just transfer your data in and it records everything that is going to be regulatory compliant with the regulations. And it does not convert your data to any arbitrary units. Another thing is stability indicating assay. You can look at not only your aggregates, your degradation products, but also what's happening to your capsid species. And that is the power of AUC when it comes to that. Process development, again, from cell harvest all the way down. We can tell you and track your progress as you are going from your cell harvest to your first affinity step, your cesium chloride steps, to all your other process development steps to enrich in there. Changing your ITRs, we can look at that. And how it affects your distributions, we can look at that. And finally, comparability, we can do that. So it's batch to batch. You should have the same S values. You should have a relatively within 10% of your CV for your quantitation and maybe 5% for your S values. That batch-to-batch comparability, we can tell you're making the same same product over and over. And this again can be in GMP. So we have to think beyond empty capsids for these, beyond percent capsids. There's much more we can do that really gives us a handle on the data. And finally, this is one of my second to last slide. How can you do it? So, what I say here is that in your initial passes, you're seeing your orthogonal modeling, noise evaluation, and your experimental design. This is for characterization and process development. But when you're starting to transition into the GMP, you think about your 21 CFR, your method and experimental optimization. These bins get a lot smaller. You have to explore a little bit more spaces in there. So explore your speed, explore your concentrations, see what it's doing on the different optimization technique, experimental parameters. We at Bioanalysis have perfected a method that is really, it can be applied to about 95% of every every sample we see. So we know exactly how it's going to behave, how it's going to look, and what information rich we can do. And just by doing that with our in-depth analysis, we can predict those speciations that you will see at higher speed. We have confirmed it. We are pretty confident in what we do with that. So your final CFS distribution, your final whatever distribution you choose to use in this case, should be GMP ready. So this has to be one of the things we want to make it when we do our analyses and move into the QC world is robustness. It has to be robust and it has to be simple. And that's how you spend the time in the method development and then transition on to the QC. At that point in time is click, 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 answer. Anyway, so that is my, that wraps me up. And if you have any questions, please feel free to ask.
Thanks for the presentations. And we have a number of questions for our discussion. So I'm going to start with a question from Carl Deal, actually my previous postdoc at Saromex in Lund. And it's a question for Kan Chen and NMR. So how would you use it? How would the 2D proton carbon method be applicable to MAB, FAB, or FC samples where methyl peaks are not designed and or heavily overlapped?
Oh, okay. So as long as the T2 decay suffice, you can use them as long as you can acquire spectra with sufficient T2 lifetime. You can use for fullness FC, FAB fragments. I'm a little bit concerned about the fullness because the T2 is relatively short, but FAB and FC are more confident to use methyl HSQC. Yeah, in case of the assignment, you really don't need assignment. They just label the peak as A, B, C, D. You don't really need to know their individual correspondence. If there's overlap, yes, you can still assign, right? You can still just say this is a class A, this is class B. You can do the position comparison and intensity comparison as well. There's no need to worry about that. Does that address?
Yes, thank you. Over to, I will get back with more questions. Lake Paul, AUC. The question is from Nicholas Larsson. Is an Excel sheet integrated in the CS distribution enough for CFR Part 11 compliance?
Great question. So when you're doing these methods with an Excel spreadsheet, you have to validate that Excel spreadsheet also. So that means your integration, whatever functions you're doing, you have to make sure those are correct. How we do our Excel, we create a custom integration program. So it's either you do the trapezoidal rule or whatever you want to do with it. But then we validate it as basically what you're doing is a CSV, okay? So you validate that Excel spreadsheet. And that Excel spreadsheet, has to have all the traceability functions that you have in there, okay? So you can custom make that spreadsheet for that.
So just a short follow-up on that from the same person is that, are you still fitting the data using ZFit for the CS distributions?
Yes, you are. So you can mitigate, you are still fitting the data, but what we do in our Excel spreadsheet is that we custom make the integration and we also report the weighted average. So we have a custom equation in that. So we're not doing peak heights like most of these other chromatography programs, which is incorrect, fundamentally incorrect, unless you're doing a GFS, which is a different type of analysis.
Okay. Risto, over to you. In the refolding assay you're using, is the denaturant formulated in the original MAB formulation?
Yes, you can basically use the original MAB formulation, or you can use any formulation you like as long as all the excipients are soluble in the high denaturant concentration. So when you use 10 molar urea, sometimes you might have solubility problems if you want to put a lot of sugars and other amino acids. So sometimes you might have to go to 9 molar urea, but then you have to check that really the antibody is unfolded in the assay. But potentially, as long as there are no solubility problems, you can do it with any buffer you like.
Okay. So it was actually a follow-up on that too, asking that the assay you just described in your presentation, is that something that could be possibly used to screen formulation components?
We actually started it as a formulation screening project for prediction of which formulations will suppress aggregation during storage. So we could see that the formulations that facilitate refolding into the monomer are the formulations where the antibodies aggregate less during storage, which is in a way logical because you assess indirectly non-native aggregation in these assays, the refoldability assays. But we now realize that there might be more interest in developability assessment. So that's why we moved with this now, more antibodies, more examples, and so on.
Right. So over back to Kang-Chen, two questions here, and I will put my interpretation on it too. So obviously when it comes to comparability and similarity exercises with any biophysical method, the qualification criteria are the key. I mean, essentially what is similar, what is not similar, right? And you have to find a balance between observing real differences and not differences that could be normal variation. So the first question is that how do you establish relevant acceptance criteria for applying the NMR chemometric method?
Yeah, so that's the reason we use the marketed drug product that's been approved recently. Right? Now we know realistically that's a similarity measure we can derive. We don't hypothetically manufacture a product, say, okay, these two need to be matched. We use industry practice, right? Industry, they already have this practice. They get approved and they're marketed, they are safe. And we take these two, they are both safe and effective. And we see, okay, how similar are they? And here we come up with 3.3 as a similarity matrix. From these two approved, marketed, safe, and effective medicine, then we say this is the similarity bar. Even though maybe is it really above it? Is it become not safe? That's another question, right? If we don't do clinical study, we don't know. But 3.3 is the kind of safety threshold that we can derive from marketed product. So the marketed product, the use of marketed product is the key here.
Right. So in your opinion, is NMR now at the stage where maybe we can introduce that in regulatory filings, and will it be possible to replace NMR with classical methods such as FTIR and circular diacritics, CD? What is your opinion on that?
Yeah, I think NMR definitely is higher resolution, and more people are using it. It is becoming more acceptable in the community. But FDA doesn't have we don't really dictate a method for anybody to use. As long as you think you approve your method is of sufficient high resolution, that's been that is encouraged. So I think that the best thing is we as an NMR community we keep applying it, and we find out if it has any usage on drug development, the similarity comparison, anything, and we come up with the pros and cons, and we discuss it, and now we are good. And we push it from both outside agency and we push from the outside agency, and when we review the data, we know the limitation and we know the pros, and we can make a recommendation.
Right. So a quick follow-up on that, because it relates so intimately to what you just answered there. So do you foresee that FDA will come up with a guidance document for the application of the various NMR methods?
I guess no, because it's really rarely that NMR been dictated as a methodology to be used,
right.
because a lot of generic firms probably don't even have NMR spectrometry. Imaging, obviously, you have. Not everybody is imaging. So so NMR is one method of high resolution, but I guess there's only one product happening that specifically says 1D NMR. You need to use 1D NMR to do the quality control, but not as I'm aware of for other methods. We dictate NMR as a method. Even like mass spec, we even don't dictate mass spec as a method. We just suggest those quality attributes need to be characterized by state-of-the-art technology, right? You can define what is the state-of-the-art technology.
Okay, thank you. Over to Lake Paul. A question from Will Rice. So he says, great talk. The value of AUC as a characterization tool in process development and comparability is pretty clear. When we move to testing, he wonders if by analogy to therapeutic proteins, aggregation, should we think about using AUC to validate the performance of other methods rather than moving AUC into the QC environment?
Will, that's a great question. I think we should both. You can validate other methods, and it's been done, as we all know, Will. It's been used to get to validate your SEC methods, usually for your antibodies. Here's what it is. I just think the next step is bringing this into the QC environment. So it should and always be used as a orthogonal method for anything, or a validation method for any type of particle characterization within a certain range, of course, and if it is well defined. So yes, I completely agree with you. It should be we should do a lot of validation, but the next step is to push it into the QC, and we need to focus on that, because it's bringing it out of that realm of characterization into what it should be done. So I'm hopefully with the gene therapy and what we're doing, and just to be very clear, this has been done in the QC environment. AUC has already seen progress in the QC environment, okay, especially in the gene therapy field. So this is not new. I just want to make sure it is there and done right.
Thank you. Over to Risto, or back to Risto. The question is, did you find any correlation between extent of protein reversibility upon heating and colloidal stability of intact proteins at ambient temperature? So, for example, looking at factors like diffusion interaction parameters, KD, and similar parameters.
Yeah, so not on a large scale. So for few conditions and a few proteins, we did measure also the interaction parameter, KD. And formulations and proteins where the KD is positive or it's higher, have higher refoldability. So in a way, things are connected. I think this is not a big surprise. That if you have high colloidal stability of the protein in the formulation, that it will tend to stay monomeric even if it unfolds. Even some people measure or try to measure this KD and also the B22, those second osmotic viral coefficient of partially unfolded proteins, which is also a great experiment as long as the protein is not aggregating during the measurements already. But there is a connection, and the question is what is easier to make, because for the KD you need very good DLS measurements, you need more sample. There are certain limitations. Also, excipients affect your DLS signal at lower protein concentrations and so on. So, yeah, there is a connection between both from what I've seen.
Yeah, thank you. Let's jump back to Kang again.
Oh, okay.
So, as we know, there are many new monoclonal antibodies going off patent soon, and several have already gone off patent, obviously. And the question is, do you expect that this value report in your presentation, 3.3, I think it was the magic number, do you expect that to be determined for insulin and rituxan? Could that be used similar to other monoclonal antibodies? And I guess we're back a little bit touching upon the qualification criteria. So in a way, do you need to qualify every new product from scratch, or could you somehow establish universal qualification criteria?
Yeah, so yeah, that's an interesting question. First, I'm not speaking for the agency, just speak from my own view. I think 3.3 is not a very strict number. It's a very general number, as I show you in the slides. The metabolomics community use a value less than 1 as a similarity. And we use 3.3, that's a very general number. It's not a very strict criteria. And I expect monoclonal antibody spectra to be similar if they are biosimilar. Because actually when you use NMR for monoclonal antibody, this large molecule, the noise is very broad. So it's effectively will become a low resolution spectroscopy. It's not as high resolution as we usually think for NMR because the noise is so broad. And also the 1D spectra only characterize the high order structure in the profile sense. It's not really down to the chemistry. So all the quality attributes that you extract from like peptide mapping or MLM, like lysine cleavage or the glycosylation, they don't really reflect in the 1D spectra because the population chemistry change are really tiny compared to the structure broadening in the 1D spectra. So I think the 3.3 value will hold. I think it will hold easily.
So since I was the one who asked this question, I just want to elaborate a little bit more on it.
Yeah.
Do you envision a point where we will get, after we have done so many qualifications and so many studies in the future, do you envision we get to a point where we say, okay, this is what was done for so many monoclonals in the past, any new monoclonal biosimilar that will be developed has to meet this criteria, or that's not really?
So again, I speak for myself, not the agency, and I don't think we will have a very strict. I think everything, every similarity approval depends on the totality of evidence, right? Maybe one day you get a value of 4. Is it not biosimilar? No, right? You need to look at all the data from all the aspects, bioassay. So NMR just give you one dimension to look at the similarity, but it's not the only dimension.
But then I need to follow up on that question again. So this is because it is really interesting. I mean, if you strictly look at the characterization experiments, comparability or biosimilarity, you don't look at assays. You don't look at any other attributes. You're looking at structure, secondary structure.
Mm-hmm.
Yeah. So would you? I mean, and I think back to both the question from George and I is that would you see that? I mean, we need to establish criteria, right, to say similar or not similar, right? So would you see that would happen start from scratch for every new product? Or do you think we can establish some kind of general threshold? I think that's
I think that. So that's something that industry can propose rather than us.
Okay.
Right? So you can propose.
But from a scientific point of view, not an FDA point of view.
Right.
So if you don't speak from an FDA perspective, from an NMR perspective, how do you see that happening? Will it be universal or will it be product specific?
I think the value of 3 or 4 might be universal. If it's really above 3 or 4, I would say your formulation is quite different.
Okay.
Yeah. That's my personal opinion.
So formulations, and a question to Lake Paul. That was a good, thank you, Kai, for bringing formulations in. So, Lake Paul, the question is, how do you evaluate S value changes in different buffers? And would you suggest doing sample dialysis to a common buffer like PBS rather than using formulation, a standard buffer for AUC rounds, or can you accept different formulation conditions?
Well, this, AUC has been around for almost 98 years, and much smarter folks than I have already thought about this problem. You can mathematically correct your buffers. So basically what you do is you convert your S values in different buffers given the different viscosities and densities, and you convert it to S20W at 20 degrees centigrade. So water and 20 degrees centigrade. So that is how you standardize the S values. John Philo's Sedentherp 3 has it's a much more comprehensive database, and that's freely available to your system. So I would, if you have different buffers, you don't have to standardize. You don't have to change it out or anything like that. You can mathematically correct for that, which is wonderful. So again, this has been done many, many times, over the years, way before I was even born. Okay, so you can do it. And to do a standard buffer, yeah, I mean, you always want to have a good buffering system. So like reducing your viscosity. But if you cannot get out of that, then, there's a mathematical way to standardize your system. And that is it's a very simple equation. And you can do it in any of the programs, or you can do it by yourself, by hand. So it's very, very, very easy to do.
Thank you. Risto, back to your experiments where you compared guanine hydrochloride unfolding, and it seems to that GUA unfolding have a weaker correlation to terminal degradation than urea-induced. Why is the degradation from guanidine hydrochloride different from other degradation mechanisms?
So there are, I think, few explanations to that. Like the one is that the buffer in these studies was like low ionic strength buffer, 50 millimole histidine. So urea usually does not change the ionic strength. So if you have some electrostatic repulsion, this, is still there even when the denaturant is inside. When you put in guanidine hydrochloride inside, the ionic strength is so high that you basically screen such electrostatic repulsions, and then you might mask some stabilizing effects. The other reason is that antibodies are in general very stable and sometimes are not completely unfolded by urea, even in very high urea concentrations. Guanidine is much more stronger denaturant, so one reason why probably urea correlates better with thermal denaturation experiments is that it not only captures, what happens with the partially unfolded protein, but it also captures a little bit conformational stability. This means some of the antibodies, they probably don't unfold completely, even at 9.5 molar urea. And usually we see that these antibodies also have very high melting temperatures, very high non-reversibility onset. So there is in a way you can think about it, there is a correlation between the apparent melting temperature and the melting denaturant concentration, especially when you do it with urea. So
would that change if you had DTT or TSEP or something? Well, would you drive that further down?
Yeah, I mean, from experiments with antibodies, from old literature, we know that if you put DTT in an antibody domain, usually the melting temperature is about 10 degrees lower. This is just because you lose the stabilizing effect of the disulfide bond. So as soon as the protein unfolds a little bit and then the DTT attacks the disulfide bond, reduces it, and then, the protein unfolds easier. Now with antibodies, I didn't do it with the DTT because, I mean, if you also lose the disulfide bonds, probably it's going to be just a mess inside. And also you don't expect, I think, from formulation point of view and developability assessment, I mean, don't expect disulfide bonds to break like during storage and formulation. So in that sense, it's very different from the refolding experiments that you do from inclusion bodies, for example, to purify protein. So it's just for different purpose.
Yeah. Kanchen, back to you. So the question is, if you would advocate for the process of using, for instance, marketed or commercially available drugs and their biosimilars to establish benchmark for orthogonal analytical techniques for higher structure assessment.
So I didn't get it as a promotion. What do you mean? What is
so the question is essentially, I mean, you used marketed drugs and biosimilars to see the variation, what is essentially similar or different from commercial products, innovative drugs and biosimilar drugs. Could that be for NMR? So the question is, could similar approaches be used for other biophysical techniques to establish essentially what is similar or different?
Yeah, I think that that's a clever way. Yeah, you can. I think you can do that. I mean, but for other methods.
And what technique would that be then? What's a follow-up question on that? In addition to NMR, I mean, do you see this kind of approach to be used as benchmarking other analytical biophysical techniques?
The method I can think of is the CD, which usually has a large variation depending on who is doing experiment, how the dilution was performed. I think that's that it is possible to establish similarity using similar approach. But it all depends on the practice. But here, I think I strengthen the drug product. If you deviate away from drug product, something unexpected might happen to change your structure differently, right? So most likely, it probably will not differ. It will make things more common to each other. Right, so then you will evaluate if this is caused by the dilution buffer you are using, because you, for example, you use a PBS buffer uniformly for everything, then the difference is gone. So in that sense, this approach will mask some differences. So if you want to do the drug product to product comparison, yeah, you can use the way similar I'm doing. But if you are not, you are doing a lot of dilution, then basically this criteria dissolved.
Thank you. Like Paul, a practical question here. I mean, we mentioned CEDFIT earlier. So the question is, why do you prefer CEDFIT to other programs? That was the first question. Is there any particular reason? Is that driven by your specific assay type needs, or is it just a preference for historical reasons?
I'm agnostic, okay, when it comes to programs. I use whatever is there. So I like Ultrascan, I like CEDFIT, I like DCDT, setting out, whatever programs I can use to do it. However, the regulatory bodies have seen CEDFIT and that distribution. So we are going with that for now. Okay, it's as I said before, that's the way it's presented. It has both a negative and a positive to it. So it looks like a distribution, but HPLC chromatogram, but it's really not. So I always got to tell people it's not.
So people have easier to understand it if they relate to something they know.
Exactly, but then you have to walk them back
Right.
a little bit. So it's all about the visualization because 99% of the data, and given the complexity of the AUC and analysis at times, you want to simplify it a little bit so everyone is general. It can really be interpreted, and that's what we do. So again, I'm agnostic. I use Ultrascan when I really want to fine tune my shapes. I use SetFit if I want to do some other programs. I use DCBT when I really want to interrogate the raw data in a model-free program. If I want direct boundary analysis, I would use SetAnal or SetFat. I mean, it's whatever the tool that you have available, whatever program you have available. I am agnostic,
Right.
so that's how I.
So, Risto, we have a couple of questions for you. I will start with this one. So the question is, in the non-refoldability DSF experiments, what is the nature of the protein after, the second baseline or transition rather? So are they composed of a mix of folded and unfolded states, partially folded states? I mean, what do you have there really in your solution?
For most antibodies, you have aggregates.
Right.
So for some, you have monomers which are misfolded in a way. Spectral properties change. But for most antibodies, it is aggregates. The good thing is that even very, very small aggregates, like which you see just as a small change into the hydrodynamic radius in DLS, because we compare it also this. Also, such aggregates are, seen as a non-reversibility because, I mean, just the tryptophan environment has changed. Even if you have like some dimers forming, especially if there is a tryptophan at the site of aggregation, you see this as a fluorescence change.
Okay, thank you.